Our next speaker is Dr. Araz Marachelian. She's the medical director of the neuroblastoma MABG program and attending physician in the hematology, oncology, and blood and marrow transplantation unit. She's also assistant professor of clinical pediatrics at the Keck School of Medicine at USC. Dr. Marachelian completed her pediatric oncology fellowship training at CHLA with an emphasis on investigation at the New Approaches to Neuroblastoma Therapy Consortium. She also obtained a master's in clinical investigation at USC. She's the medical director of the neuroblastoma MIBG and also the medical director of the operations of the Nant consortium since 2010. She is involved in protocol developments at Nant and oversees the conduct of the studies and their completion. Her research emphasis is on studying new immunotherapeutic approaches to the treatment of neuroblastoma, as well as in studying minimal disease detection. Dr. Marachelian chairs clinical trials within the NANT consortium and leads the post-consolidation task force and children's oncology group. She will be presenting today updates on the NANT trials and some study results from previous trials. Dr. Araz Marachelian.
Where is mine? There we go. I'll practice this time. Thank you so much, everyone. Thank you, Pat, for inviting me again, and I'm really looking forward to giving our update again this year. And maybe I'll stand right here because I may not be able to see my own slides. Some vertical issues. Okay, here we go. We'll start with our mission. Our mission, I think, is simple and bold. We're saying to cure all children with high-risk neuroblastoma. How are we doing that? Using novel precision therapies developed by our expert network of the laboratory and clinical investigators. So what is the NANT Consortium? For anyone who's not aware, we bring together experts from across the country. We have clinicians, scientists, researchers, patient advocates working together for the shared goal: better outcomes and access to children with neuroblastoma. This is a picture at a meeting. We need another one that is actually maybe a little bit better, beautiful background, but next time. I think our next meeting will be in Philadelphia in the fall, so we shall see if we can get some foliage. All right. So the NANT Consortium scientific chair is Julie Park, and I'm the medical director. And there's a nice group of investigators that work all together. But how did it all start? It started with a program project grant. This is a multi-team NIH grant that supports long-term collaborative research to tackle problems in neuroblastoma, like treatment resistance. So generally, PPGs fund things like concepts that have unifying scientific goal. You need to have disciplinary collaboration, and you need to have integrated lab and clinical science together. So the PPG partly and only partly funds several lab projects and NANT, which turns discoveries into clinical trials. The idea is we're taking things from the laboratory over to patients. Together, we're studying really how neuroblastoma evades the immune system and how to stop it. And I know the question that comes up all the time: how is the current climate affecting us? Yes, there's some effects, but diversifying your portfolio is also always very important in terms of your ability to continue the work that's absolutely needed in our children. So this is just historic, but since we always talk about the consortium, these are the dots of where the NANT sites are. It started off in 1990 as 8 sites, and now there's 18 experienced children's cancer centers in North America and Europe. And the number of patients who have enrolled is about 1,400 or a little more. So we're really proud of that. And we try to, have geographic locations where patients can access. This may be missing one site, but you can see what's also very important is that we now have these transatlantic collaborations. We always had Canada involved with our trials, and have then extended it to Europe, and have now Princess Maxima helping us as being the Nant operations of Europe, so that we can better get studies in Europe and quickly. So before, really this is the pipeline, lab, early phase trials, involvement of everyone, making sure you have an infrastructure of biobank so that we can eventually get to the rationale for larger studies and translate the studies. I only have that picture up to show that even though it's supposed to be a straight line, it's never a straight line. There's a lot of ups and downs, and really that is what I'm going to show for the next little bit. So what I'm planning on doing is telling you a few stories where we're going to talk about a few agents. Some of you might have heard part of this, but I've added a little bit, and what you may hear parts of this in some talks, but I'm trying to give you this overall story of how we're putting this together. So we'll first start with the lorlatinib and the MIDA development. I think you're going to be hearing Esther Berko talk a lot about circulating DNA, but I wanted to just show you how, an idea can go forward to, a phase 3 trial where it was identified that a diagnosis, 10 or 15% of patients had an ALK aberration, whereas in the relapse refractory setting, this was a higher number. At the same time, the ALK mutation identification, lung cancer developed lorlatinib as another agent that could overcome resistance that we had seen with crizotinib. And so preclinical data at the Mosaic lab occurred, and the Araz lab, who actually is also the scientific PI of the program project grant. And the work had happened, and we were able to look at that data and say, great, lorlatinib seems to work much better than crizotinib. It works in neuroblastoma patients. Let's bring it to patients. The trials occurred. 1502 means the first concept, 2015. But you can see there promising results of 30 to 67%, and moved over to phase 3 trials. I will show you, since I'm going to do this again later on, these colorful graphs. The ones that are going down means it's a good response. You have a Curie score decrease. The ones going up are Curie score increase. You can see generally it's on the way down. The adults seem to do better for some reason, but there's a lot more that we need to understand. As the phase 3 trial is ongoing, we have also been looking at, how did the response correlate with what we would see in circulating DNA. And you can see they're kind in different ways of progressions, which is really important to understand. So in the first graph where you see those red dots, those are patients who have progressive disease. And you can see the bigger the red dot is, the higher amount of ALK tumor circulating, essentially. And so you can see as the red dot occurs, it was bigger. So that means those patients had an ALK mutation that now was resistant because it was growing despite lorlatinib. Whereas the other column, you can see the patients are progressing, but the dot is not getting bigger. The other ones, you don't even see the dot, you don't see the ALK mutation. So all kinds of different kinds of patients, and we really need to divide these patients and understand them better. And that was the work that occurred in the Mosaic lab with Dr. Berko as well, looking at what are the new mutations that are coming around, and then how did these new mutations become resistant. So I'm not going to take away from Dr. Berko's talk, but the idea is to do a clinical trial, but really learn from it as well, and see how we can improve our next trials. because if we don't understand resistance, we don't know what to do for our next trial. Which brings us back to biology. So the biology study in Nant has been around for a while because it's 2004. It's a rich repository of relapsed patient specimens and could be requested by any scientist with clinical annotation, blood, bone marrow, tumor, stem cells. There's scientific review. In fact, I'm excited about the fact that the bone marrow on our patients that have tumor have been used recently on some projects looking at expression of GD2 and other expression. So this is a valuable resource that we continue to expand. The Nant Precision trial was a trial that occurred with the Nant biology, where it was our first attempt at looking at biopsies and looking at the correlation of biopsies of tumor and the types of mutations that we may have and the type of chromosomal aberrations that we had. This graph is only to show us all that these things don't occur just randomly. So if there's a patient who has an ATRX mutation, they, for example, are likely not to have an NMIC amplification. They're combinations, and each one of these combinations tell us that there's a different kind of disease and a different kind of targeting that needs to occur. Of course, we can't break down neuroblastoma into 50 different types of neuroblastoma, but these are the first attempts at really looking at how can we divide these tumors. So what we are doing then is taking all this information. We have realized that we really need a consistent way to look at cross studies all our molecular and immunological characterization. So that's how MIGHTY came about. We have in the past gotten molecular characterization or immunological characterization of different studies or patients, but really to have it centrally and to do it at multiple time points so that we can see tumor evolution, whether it's in mutation and resistance or it's in its cytokines and immune cell differences, etc. And so this MIGHTY integrates this multi-layer data in hopes of personalization of therapy, but really improving that. And like I said, we can't have 50 different trials for 50 different type of neuroblastomas. And so really trying to understand what is the most important thing, how can we predict either relapse early or resistance early, and or, which patient should get which therapy. And this, of course, then shows the ultimate goal of MIGHTY is to inform new combinations, new ways to hit the tumor harder, smarter, more specifically, and laying the groundwork for next generation of therapies. And so all patients who enroll on the MIGHTY will be able to get all these fancy circulating DNA and all the immune workup. Some of it will be returned back to the patient, and the rest of it will remain as a research study. We expect to open MIGHTY very soon. So now that we did a little bit of this biology, I'm going to switch gear to current therapies. So the first one I'm going to talk about is MIBG. I think many of you know that MIBG has been around for a while. It's concentrated in the sympathetic nervous system with the NET transporter, which I call the elevator. It's really the original targeted therapy. And it, of course, has moved to COG studies already, and we are awaiting those results. However, we still need to work on improving that response rate of MIBG when given in relapse refractory setting. So I'm going to tell you a story of how consecutive studies helped us get to where we are and where we want to move forward to. So first off was the vorinostat addition to MIBG. So the first study actually was done, this was chaired by Dr. Du Bois. It was a randomized phase 2 study. So there were 3 arms, alone MIBG, the other one is irinotecan and vincristine, and the other one is vorinostat. And vorinostat came out ahead and it was safe, feasible, and quite active. It set the stage for more combinations, and you can see again back to the graphs, the curves on the way down. You can see the vorinostat arm having a CR, PR, MR rate of 41%. Great. So that became our standard for MIBG for treatment. Next was to look at if we can add other agents that could be active. So combining the two most active therapies in neuroblastoma, which are GD2 as well as I-131 MIBG. This idea came also from the fact that there's a known, I love the term, abscopal effect, where a radiated site in one spot triggers a response to immunotherapy in other spots. And so this is a well-known phenomenon and also seen in adult tumors. And so the idea was if we can combine MIBG as that radiation and we have immunotherapy on top of each other, then we could potentially not only have the response of whatever the response of the agent, but also add to it this abscopal effect. During this time, we also understood the vorinostat also increases GD2 expression and immune cell infiltration. So even though initially vorinostat, in our thought, was just a radiation sensitizer, this was added. And so it can potentially overcome resistance. So the study looks like a long list, but really it's MIBG, vorinostat, like we said, and then there's dinutuximab cycles on their own. And that was Nant 1701. I'm happy to say that we just got this published or accepted for publication. Nant 1701, the overall response rate of CR, PR, MR, was 55%. And you can see here when you look at the first graph, those are the patients who were in a part of a dose escalation. And you can see there are different dose levels there. And that was just MIBG and GD2, dinutuximab. And then the next one, which is part B, is when the addition of vorinostat occurred. And we can see the response rate being 75%, which is, of course, very promising. But we need to, it's a small number of patients, as you can see there. And so this needs to be confirmed in a larger setting. Great. We are very happy. Dr. Thomas Cash actually got this to goal with the publication recently. So the next, natural idea is then to say, well, we have MIBG vorinostat, we have MIBG dinutuximab, and can MIBG dinutuximab and vorinostat all combined together. This is the 3-arm study, and we are actively working on its launch. There was you can see the number is 21, and it's not yet open, but that's because of some drug supply issues for a while. It looks like we are overcome most of the drug supply issues, and we are working actively on a launch and perhaps just some contracts, etc. And of course, our advocate committee did help us in that job. So you can see here the years. Whenever I'm saying 11, 17, 21, it doesn't mean that the study started then. It was just really in early concept form. But as the years go on, we keep on adding and learning. And our hope is that the gaps in between these years are not that long. And that is ultimately the goal of starting another study immediately after the next so that we can learn and we don't have patients who are being treated on compassionate access, for example, and we aren't learning and moving the field forward. Okay, so that's MIBG. Let's talk about chemoimmunotherapy. We're combining chemotherapy and immunotherapy. Obviously, this is very familiar to everyone in this room, I assume. There was the trial in COG initially that showed improved outcome for relapse refractory with that 40% response rate. And it was really a turning point for us. that adding dinutuximab to relapse chemo improved responses. And really, from there, we need to figure out how to improve that. 40% is great, but we have a lot of patients who may respond and don't respond completely, or patients who respond and then progress later, and or all kinds of parameters of this. And so we know that it works, but we need to make it better. So there's a lot of work ongoing in making it better. And so the next little few minutes, we'll take the little tour of how to make it better. So number one is the idea of improving immunotherapy with natural killer cells. What are natural killer cells? They're just these great part of the innate immune system, meaning they're not like T and B cells. They don't need to get information before to fight something. They're just ready all the time to come in and kill whatever they think is wrong. And so you don't need that prior exposure, so it makes it really nice. So they're good killers. They're, the picture's cool and it's real. It's not just it's not a fake drawing. And they come, they release the molecules, target cells. They really normally are supposed to come in and kill virally infected cells, cancer cells, stress cells, any cell that looks wrong. And they produce these chemicals to signal other immune cells to come and help them. And they have a short lifespan. So they're there, they do their job, and they die off. So they don't have that memory that we're talking about. So we need them in, and they go out. My little very rough drawing of how we need to make things. So number one, we can think about ways to give patients NK cells, somehow make their NK cells better by giving them a number, or we can give them chemicals on the other side to make them more active. So we'll talk about both of these strategies and do a little history again. So number one, the first trial was 1301, and that was when we decided to take the NK cells, expand them from the patient's neuroblastoma, the neuroblastoma patient's blood. And then we grew them, and we gave them after dinutuximab, and this was pre-chemo immunotherapy era. We learned that patients did great. They had no problems with the NK cells. We knew how to dose it, but so it set the path forward for other trials in neuroblastoma and other diseases to use cellular therapy. And I'm a huge proponent for multi-institutional cellular therapy trials so that patients have access all across the country. And set the stage for our current proposal, which I'm going to talk about. Limitations are we have to wait for the NK cells to grow. It was only 14 days, but every day adds for our patients. And the NK cells from neuroblastoma patients, surprise, surprise, were not great in number because they were treated with chemo, etc., or function. They come around and they're not the best killers that we can imagine. And of course, there was cost of individual NK cell production. So the trial was done. It is not yet published. It is coming. We are working on data. So what can we do from there? The next idea was, we need to make these NK cells better. How can we make them better? Grow them around things that look like neuroblastoma environment. Neuroblastoma is really smart, releases things. One of them is TGF-beta, which is an immunosuppressant. So if you grow these NK cells with TGF-beta, they are stronger. And so that's called TGF-beta imprinting. I'm going to have little ducks walking around next time. We have great killers because they're around TGF-beta. So how to make universal donor NK cells. Here's the little formula. Find the donors in bone marrow registry, which have been found. Seek donors with strong NK cells based on their biology. Many of us have, some of us better and some of us not so great NK cells. We collect them, grow and expand them, imprint them, so meaning grow them with TGF-beta. And now we have these ready to use, what I call supercharged allogeneic NK cells. Great, they're off the shelf, And now here's the trial where the patients are enrolling. They get chemoimmunotherapy the regular way that many of you may be familiar with. And then following that, on day 8, when there's still lots of dinutuximab and immune system going on, the NK cells are given. So the trial is supposed to be 50 patients at all 9 institutions. It did open. It is a little bit of a pause because we are looking at production changes, cost issues due to production, and trying to make sure that we have enough of the money to be able to get all our 50 patients. So that is why it is currently paused, and hoping in a few months we'll be able to open it back once we are completely set. So that's the active trial. But how else can we this is to give cells. We talked about activating cells. The trial that we did before was adding lenalidomide to dinutuximab, and I just put these curves down there just so you can see, not because I'm going to go over them, but when you give things like lenalidomide with dinutuximab, a lot is changing in the patient's body. We did these at quite often intervals where we look at their T cells, B cells, all kinds of things. So lots happening. So you will hear a little more. There is follow-up on these. There are 2 different other ways to activate that are coming, the IL-15 superagonist and the iberdomide, and both of these are going to be at either COG, St. Jude, but it will be available for all sites. So each building block, as I said. And finally, we need to remember that NK cells are just one piece of this equation. We're looking at other immune checkpoints like TIGIT and PD-1 that can suppress immune responses. If we can block those pathways, we may be able to unleash even stronger anti-tumor activity. So the thought is perhaps doing a chemoimmunotherapy trial with TIGIT and PD-1, but that is still in the works. So you can see lenalidomide, the auto-NK cells, the allogeneic NK cells, and possibly targeting TIGIT and PD-1 as potential trials coming up. So, last little story is figuring out neuroblastoma response. To test therapies, we need better ways to measure response. And that's been a major focus of our work, not just tumor size, but understanding how the tumor responds, whether it's active, and how to quantify it. So this was the work that actually led to the international response criteria. We're looking at Curie scoring, delineating differences between cold tumors and active tumors, and we continue on really having very focused and deep response reviews on our patients so that we can continue to inform this field. In fact, we are looking at this again to look at in the current era who's at risk for progression, where do they progress, as well as looking at differences between different patients, whether they, how do they respond based on what they have, and where do they progress, etc. And of course, the MIBG is where we are right now, but we need to look at other nuclear medicine modalities, and the MFBG study that was done that is also being presented at ANR had a one-time comparison to other evaluations, but of course need confirmation in serial assessments, which is ongoing or planned in COG. Of course, there's challenges. We always have drug supply issues. And, despite the fact that I'm talking here for a while, giving you results and histories, this is just, we do recognize that we need more trials, but we only can do more trials if we have industry partners who can work with us to be able to bring some of these agents forward. Of course, there's still things to overcome, like high therapy costs, and tumors do evolve, but we can evolve too. So there we go. So every trial shapes our future care. This is what keeps us moving forward. There is a story about each trial and one building block over the other. And so each sample teaches us something new. Each family shapes the future. And of course, it's the collaboration, whether it's scientific, industry, advocate, all of it together so that we can make this happen. And I wanted to just thank you all for listening to my multiple stories that hopefully have some links with each other. And I'm hoping that we can see more trials open soon. And thank you, everyone, for enrolling your patients on any trials. And without our supporters and funders, we would never be able to do this work.
Thank you, Dr. Marachelian. Does anyone have any questions? Got one coming.
Hi. I was wondering, has anyone done any sort of comparison between the strength of NK cells in low-risk neuroblastoma patients compared to high-risk neuroblastoma patients?
I'm not aware of that. There's been a lot of work on the overall immune milieu, but I'm not exactly sure specifically the data for the NK cells. I think in low risk or non-high risk disease, some of it is the immune system, but some of it is also what's happening with the sympathetic nervous systems, natural maturation cycles. But I'll take a look at it, and if I find something, I'll let you know.
Because it just occurred to me that perhaps that is a significant factor in why those low-risk neuroblastomas are low risk. Perhaps the strength of those NK cells are providing so much immune power. And maybe, just maybe, you use those patients to create your NK cells.
Yeah, I mean, I think part of it is, with the non-high risk or low risk kids, most of these are occurring in the very young children. I almost feel like it is possible, but I feel like if I thought high risk neuroblastoma was 50 diseases, this is very different. It's almost shouldn't even be called neuroblastoma, just because of its. I think it's. Obviously, that's just an exaggeration, but in reality, everything that's happening in it, in terms of maturation and apoptosis, seems to be a very different mechanism than what's happening in the high risk.
Hi, thank you so much. I saw on the slide the lorlatinib is a drug used for adults, initially for lung cancer. Do you know the percentage of orphan drugs that the kids are being treated with? And can that help offset the huge costs of finding new drugs, finding new partners? Like when my son was going through treatment, I know we used a lot of orphan drugs. Is there a percentage?
I don't really have an exact percentage. One of the things that we struggle with, for example, there are other drugs that are FDA approved. So we would think, we have preclinical data on some FDA approved agents that look very exciting. However, if we want to combine it in a clinical trial, insurance is not going to pay for that drug just because we have preclinical data generally. And so unless the company is interested in giving it to us, then we can't really do the trial. And so this is where we are stuck a little bit sometimes. And if the timelines for the industry partner don't really match up in terms of their what's called pediatric exclusivity, where, the patents, et cetera, then they might not be as interested in providing the drug for us. And that's been a challenge. But I do agree there are situations where we've done some trials where we've used FDA-approved agents with some minor data, and I think that is where we have to go at some point, especially in agents that are not as expensive. That's a little easier, but when the drug cost is really expensive, then we're really looking at insurance covering it, and it could be very variable and can be also not fair in terms of one patient's insurance versus the other, and we really want to have access for everyone.
Okay, thank you so much.
Okay. Anyone else have any questions? Okay. Thank you, Dr. Marachelian.